Welcome to this class. In the last class, we discussed mostly about the electron microscopy, transmission electron microscopy, and also we have covered the diffraction and also the imaging. Now in this class, I will introduce briefly to the scanning electron microscope, and we discuss about the image formation. Then we will introduce to the electron channeling contrast imaging. Okay, and how these techniques can be used in combination with the electron microscope, which is transmission, and also the atom probe tomography. That I will briefly, what I will do is I will present all the applications with different alloy systems. So here the SEM is similar to TEM. Okay, and both employs electrons, beam of electrons. They have an electron gun, and they have a condenser system. They have vacuum system. So most of them are similar as in the transmission electron microscope. The only difference is TM provides the internal structure, while SCM provides the surface or near surface structure of bulk samples. Okay. Okay. Now here in SCM, the electron gun can be similar to TM, can be thermoionic, can be Fexos. Okay? And the electrons are accelerated between 1 kilo electron volt to 30 kilo electron volt. Okay, so it has a series, it has a one or two condenser lens systems. Condenser lens system. The main function of this condenser lens system is to demagnify the beam of electrons. Demagnify the beam of electrons, and the demagnification after that the electron beam is around 2 to 10 nanometers. Okay, and in this the electron beam is scanned across the specimen, okay, by the coils. And while this electron beam, the primary electron beam, scans the specimen surface, then due to the interaction of electron beam with the sample surface, there will be a secondary electrons, backscattered electrons, and other radiations which are coming. And these electrons, secondary electrons or backscattered electrons, are detected by detector. Okay, and usually they actually counts the number of electrons given off from each spot. So whenever there is a specimen, if there is a beam falling on that particular spot, from that spot, the total number of electrons which are coming out of the sample surface, then there is a detector which detects those electrons. Okay, and at the same time, while the electron beam interacts, with the sample, similarly, there is a spot on the cathode ray tube. CRT monitor, what those at the same time that also is scanned across. Okay, and the brightness of spot corresponds to, can be modulated by the amplified current from the detector. Okay, as this electron beam scans on the surface, so there is also a scanning of a spot on the CRT monitor. Okay, so this process is called rastering. Okay, it has a condenser lens system, and this condenser lens system, actually the role is to demagnify the electron beam to a particular spot. And the electrons which are coming from the specimen after the interaction, it goes to the detector. And as the beam scans or rasters on the sample from that particular spot at the same rate in a cathode ray in a cathode ray tube, the spot also scans along that particular screen, and there is a amplification of that particular electrons which are detected. Okay, so that is directly related to your brightness. This process is called rastering. Okay, so this is a rastering on the CRT monitor. This is rastering on the specimen. So when a length L rasters, the electron beam rasters on the sample, or the small L rasters on the specimen, then you can get a linear magnification that is equals to L by L. Okay, and this is, so for example, if you have a 10 microns into 10 microns on the sample, and if the same spot and similar spot rastering on the CRT is around 100 mm into 100 mm, then you have a magnification of 10,000 x. Okay, so this is the scanning or rastering of the beam along the sample surface. Here I am showing you the electron once incident on the sample, then it will penetrate into the sample surface, and it has a this particular volume is called a interaction volume. And in this interaction volume, depending upon the the energy E0, and depending upon the incident, the radiations which are coming out, the energy of the radiation of the coming out, various radiations are generated as a result of inelastic scattering. And the energy of the radiation depends upon the energy lost when the primary electron beam travels into the sample surface. Okay, and these radiations are not detected unless it escapes from the sample surface, and that depends upon the type of radiation, and also it depends upon the specimen. Okay, so here, so for example, here the X-rays which are generated from the sample surface, these are actually cannot be absorbed easily from this from the specimen. Mostly these will escape because of the high energy. Okay, so here in the left there is a cross section where you can see that there is an incident beam. You will get an X-ray from the sample which is gives the composition information. You will get Auger electrons which gives the surface sensitive compositional information. You can get primary backscattered electrons. I will come to these backscattered electrons in the later. Actually, these backscattered electrons gives the atomic number and topographic information. There is a cathode luminescence which gives the electrical information that is related to your conduction band electrons. Then there is a secondary electrons which gives the topographical information. Okay, and these are the electrons which are released from the specimen atoms after the interaction of the beam. Correct? And this is your sample. So the volume inside the specimen in which the interaction occurs while interacting with the electron beam, that depends upon the following factors. First is atomic number. The material being examined, higher atomic number material absorb or stop more electrons. So they have a smaller interaction volume. Accelerating voltage. Higher voltages can penetrate farther into the sample and generate a larger interaction volume. Also, one more factor, important factor is angle of incidence. This is the greater the angle, the smaller is the interaction volume. Correct. And here you can see that approximately, this is schematic where it is shown that approximately at the depth of 5 microns, you can see that as the secondary electrons have much lower energy, it usually comes from the top surface. It is within a micron, within less than a micron. Okay. Backscattered electrons will be between 1 to 2 microns, and the range of X-rays, K X-rays and X-rays, these are within 5 microns. Okay. And each volume from which these radiations are generated, each volume is called sampling volume. Okay. So you can see that there is an interaction volume and there is a sampling volume. Sampling volume corresponds to each type of radiation which are coming out. For example, the sampling volume for X-rays are much larger than the sampling volume for BSC, than the sampling volume for the secondary electrons. So sampling volume definition, what is the definition? The volume of the material which contributes to the signal. Okay. For X-rays, you can see that the sampling volume is very large and it is almost equal to the interaction volume because of the higher energy. Okay. In backscattered, the electrons will not backscatter if they penetrate more than a fraction of micrometer. Okay. And BSC usually originates from a very small region. Okay. Now, here I am showing you a intensity versus the electron energy. E0 is the incident primary energy. Now you can see that backscattered electrons are the electrons when there is an incident beam, these primary beam actually loses energy while going to the sample interior, and it actually comes back as a backscattered electrons. Okay, so it might possible that the backscattering can occur at a very low sampling volume, at means near to the surface. So these are backscattered electrons, and these A type. So there are two types, A type and B type. A type backscattered electrons, they have a very high energy because they lost very less energy from the E0. Okay, and also they have a very good high spatial resolution. They have a high spatial resolution because they are coming from the only the top surface. However, the electrons which travel into the sample and they are coming out, they lose loss of energy. So these are the B type of backscattered electrons. Okay, so you can see that in this figure, this particular B corresponds to the backscattered electrons and it has a large spread. Okay, so have been scattered only. So the higher energy, it means that the scattering has taken place only for few times. Okay, so A has a high spatial resolution, so you will get also the crystallographic information. B has lost lot of energy, so the spatial resolution is will be very less. Correct? And one more important part is the secondary electrons. What are secondary electrons? When an incident beam interacts with the sample, what will happen? The electrons from the atoms on the sample, they gets knocked out, and those electrons are called secondary electrons. And when the backscattered electron travels, and when it comes out of the surface, it might possible that it has a sufficient energy to knock out again these secondary electrons from the sample surface. So these are Se2, and the initial electrons, these are called Se1. So Se2 are the electrons which are coming out of the sample surface or some sample atoms by the backscattered electrons which are coming out. Okay, so secondary electrons generated by both primary electron and also the escaping backscattered electrons. But however, the primary from the generator from the primary electron will be numerous, very high. Okay, and these secondary electrons usually originates from a very small area which is near to the beam size. Okay, and secondary electron has a smallest sampling size. We can quantify them with a coefficient. These are called secondary electron coefficient, which is written by delta, and backscattered electron coefficient, which is by eta. And the terminology is the secondary electrons. It means that the number of electrons, the number of electrons or number of electrons which are secondary or backscattered electrons from the specimen for each incident electron. These are known as secondary electron coefficient or the backscattered electron coefficients. Okay, and here the n strongly depends on the atomic number. So electron yield versus atomic number, you can see that for the eta, which is a backscattered electrons, you can see that as your atomic number increases, your electron yield also increases for the backscattered electrons. So eta is dependent, but however, the secondary electron delta doesn't depend upon the mostly on the atomic number. Okay, so here you can see that the backscattered coefficient with the tilt angle. So as I just introduced about the interaction volume. Okay, so as I told you, as you increase the incident electron voltage, you can see that the interaction volume also increases. Okay, so interaction volume also increases. So these are the Monte Carlo electron trajectory simulations. Okay, and with depending upon the atomic number, also. So for iron, you can see that the interaction volume is very less, but for the low atomic number, the interaction volume is very large. And also, as I told you, specimen tilt. If you see that, and this depends upon your angle phi, the angle between the incident beam with respect to the sample surface. Now, if it is at 0 degrees Celsius, you can see that the interaction volume, and while at 60 degrees Celsius, you can see that the escaping tendency of these electrons from the sample surface increases significantly. So the electron yield, or it will be much higher as we increase the tilt angle. Fine. And this is the thing which it is shown here. The backscattered coefficient increases as your tilt angle increases. Fine. Now, backscattered coefficient versus the energy. You can see that the backscattered one, which, as I told you, when the beam interacts from the top surface itself, these are backscattered. But there is a backscattered too, which travels along the surface and it comes out. Okay, so the intensity, the E efficiency coefficient of backscattered is much higher for the uh, A type. These are A type, these are B type, second type. Okay, and with the tilt angle, as I told you, with the tilt angle delta theta, you can see that as the tilt angle increases, your secondary electron coefficient also increases. Okay, so that also depends upon your theta. Now, if there is an interaction volume at the center of the specimen and the interaction volume at the near to the edge, you can see that at the near to the edge, the yield of these electrons will be much higher. And but at the center, the yield will be much lesser. Due to this issue, what will happen? You will get the edge effects in the sample. These are called edge effects. You can see that the here this region is brighter, but while the central region is relatively darker. So these are called edge effects. Now, if your sample surface is as a something related to the fracture surface, okay, so you can see that the topographic contrast, if your sample surface is not uniform, then you can see that wherever there is a dip, there the electrons, the yield of these electrons will be much less, but wherever there is a tip, then the yield of electrons will be much higher. So you can see that easily these secondary electrons are used for the topographic contrast. Okay, so with this, I will end this class now. And we have gone briefly about the SEM, and we also discussed about how there is a difference between the backscattered and the secondary electrons. Now, in the next class, what we briefly go through is the detection of these secondary electrons and the backscattered electrons.